Okay, so maybe we can get started with some introductions. I'm really pleased to welcome everybody back to this Drug Hunter Flash Talk. Today we have two guests from Nanoform, which is headquartered in Helsinki, but Christian here is based in Cambridge in the UK, and Chris Worrall is based in San Diego, right?
Yep, that's right. Thanks.
Okay. So first, maybe let's just introduce the two of you. Christian, you're the Chief Commercial Officer of Nanoform, is that correct?
That's right, Dennis. Yeah.
Awesome. And I saw on your profile you started your career in process chemistry. Can you tell us a little bit about that?
Yeah, of course. A chemist first and foremost, and started off in process chemistry and drug discovery, and quickly realized that I enjoyed talking more about the chemistry than actually doing it myself. So moved into commercial, and my first commercial role was actually in particle engineering for a small startup company in Oxford, UK, called Prosonics back in 2006. And I was the first employee and helped to sell that technology into the industry, utilizing ultrasound for crystallization. And quickly we realized actually that as a technology, that ultrasound could have a lot of implications for particle size control, seeding, nucleation, crystal growth, etc. But it could also really enable drug formulation performance. And we focused on respiratory drug delivery. And I spent 5 years there. Then I moved to Dr. Reddy's to help sell their services in drug substance and drug product manufacture to innovator pharma and biotech throughout Europe and Asia Pacific. Then my last job before I joined Nanoform was with Johnson Matthey, where I helped to set up a European CDMO offering for them. They're now Veranova. They came out as a spin out recently from through a private equity acquisition of the healthcare sector for JM. But I was there leading the solid form group commercially called Pharmorphix that we acquired from Sigma Aldrich, helping to develop the particle engineering strategy for the health sector and also driving the CDMO offering in Europe. And then I got a call from this company called Nanoform back in 2018. And I have to say, to begin with, I was a little bit skeptical because, I understood the particle engineering space. I also understood that supercritical CO2 has been around before. And, I was aware that there were challenges previously in terms of scalability and reproducibility. But I was really pleased that I stayed on the line and listened to the headhunter and spoke to Edward Hegstrom, the professor of Helsinki Uni that set the company up, that invented the technology, and his brother Albert, the CFO, and realized actually they're looking at this technology from a physics perspective to really get good control of the process, to be able to scale the technology and to make it reproducible, and being able to make particles as small as 10 to 50 nanometers in a dry particle form added huge opportunities for drug product formulation. So I jumped at the chance to really try and help this company take this technology into the marketplace. And when I joined, there was only 18 employees. We're now 150, headquarters, as you say, in Helsinki. But we have ambition to have a GMP manufacturing facility also in the United States. So we're currently looking for the right site there. We've become a publicly listed company in that time. In June 2020, we listed on the NASDAQ in both Stockholm in Sweden and in Helsinki in Finland. We've raised about 150 million euros in the last couple of years to further develop the CESS technology that we'll mention later today, and also to develop a new biological nanoparticle technology. It's been a fantastic journey, but I think we're only just scratching the tip of the iceberg as to the impact that we can have in pharma. So thank you very much, Dennis, for having us on this show.
Absolutely. Fantastic. And Chris Worrall, I understand you have a PhD in synthetic chemistry, is that right? Before joining the formulation space.
Yes, indeed. Yeah, as you said, I did my PhD in synthetic chemistry with a significant focus on atrial isomerism as well, and controlling restricted rotation. And I think, one of the molecules that really kind of got me intrigued in the pharma sector was vancomycin. Obviously, it has three restricted axes, but two of those are diaryl ether axes. And that was basically what my PhD was on, was controlling restricted rotation and atrial isomerism around diaryl ethers. And so, yeah, after my PhD, I started working in process chemistry. So it was one of those very much immediate things. So I had an interview, and I finished in the lab of my PhD on the Friday, and I started in the lab and doing PR&D work the following Monday. So I actually wrote up whilst doing the PR&D work. So yeah, I did 2 years of PR&D towards GMP syntheses. And then I moved internally within Sigma Aldrich to a company that Christian's already mentioned, to Pharmorphics. And so I spent 13 years with the Pharmorphics group. So initially in the lab, and then project management. And then 10 years ago, I moved to the dark side, if you will, and into commercial, and moved also from Cambridge over to San Diego to look after the commercial activities for Nanoform, for Pharmorphics in the US. And then about 2 years ago, I got a call from Christian saying it's very similar. Hey, we're expanding the team at Nanoform. Would you like to come on board? And obviously, everything that Christian's alluded to in terms of where Nanoform is going, what we're doing, was hugely exciting to me. And so yeah, I jumped at the chance and came over. So yeah, I'm VP of business development, based obviously on the west coast of the US.
Fantastic. Thank you. I'm curious, I guess our audience doesn't normally get to hear from people who have a lot of experience in formulation in solid form. What are your favorite molecules and why?
I mean, obviously, I mentioned, vancomycin was obviously kind of the first one that came through, just because the restricted rotation. One of the ones that I looked at quite extensively would be ibrutinib. And so there's a number of different solid forms for that, and not just in terms of the polymorphic space, but also salt forms, and then also co-crystals as well from that. From a medchem perspective, it's obviously very interesting, right? Because it was a test compound initially that the other compounds were compared to, and then Pharmacyclics then elevated that as the compound of choice. So that's very, very interesting. And then aprepitant as well, because of it took nanomilling technology to enable that molecule to go through, because of the poor aqueous solubility. But there's also potential co-crystal applications for aprepitant. And then obviously because the potential for generation of nanoparticles of that, it's obviously interesting from a nanoform perspective as well.
Interesting. How about you, Christian?
Yeah, I'd probably just add, I think there's been some interesting molecules that have succeeded to get on the market, even despite their complexity and their challenges with bioavailability. And there's drugs on the market today that only have a few percent bioavailability. And I think there's just such huge opportunity to make better versions of them. But also, when we're considering the screening from a drug discovery and preclinical perspective, to really start to implement technologies to make sure we get the best for the patient at an early stage. And one example perhaps could be everolimus. It's an mTOR inhibitor. It's used to prevent rejection of organ transplant and the treatment of various cancers. It's really poorly soluble in aqueous media, sensitive to heat and light, and has a molecular weight of over 958 or 60, something like that. A log P 5.9, and the bioavailability and PK parameters are highly variable due to the poor solubility of the API, efflux transporters, and first pass metabolism, and it necessitates frequent blood monitoring. So I think that's a molecule that's quite interesting, where if you could make a nanoform version or a nanoparticle version, perhaps there's an opportunity to get a solid oral dosage to improve the bioavailability, resulting in dose reduction, less variability in PK parameters, resulting in less frequent blood level monitoring, as an example. And I think there's a lot of drugs today that are on the market that are there, and they've successfully made it there. But we know what we shouldn't forget is all the drugs that never made it there, right? Failed in phase 1 because of their bioavailability issues. And I guess if there's an opportunity to, screen molecules early on, and in drug discovery, change the lens at which we look at molecules and molecular space, to say, well, given our sort of in silico design of these molecules, we currently would put forward X percentage of these molecules in the library to take forward as to lead candidates, but we'd discard these other ones. If we change that lens and said, actually, if we utilized a technology that we know can support these molecules, maybe it would increase the number of drugs we can take forward. And that's our philosophy. And that's why we've developed an AI platform called StarMap, which is pure in silico screening and can be used online by our pharma partners to be able to do just that, to be able to screen thousands of molecules and to identify the ones that are a good fit for our technology, so that they know that if those molecules are interesting, they could take further compounds into clinic. And I think, when we see the complexity of drugs today, we need all the solutions we can get to take them forward. So I'm kind of, on one hand, really delighted by the synthetic technologies that have been able to make these molecules so successfully, and on the other hand, enamored by the challenges that we face and trying to come up with solutions to overcome them from a bioavailability and formulation perspective. So yeah, that's my thoughts.
Yeah. That's a great segue into the topic of this talk, how drugs have evolved over time. I really appreciate both of your very thoughtful answers. Actually, I did see a question in the chat which was asking how close your company is to commercializing this technology. And this is good timing, because yesterday I saw a press release from your company that you are now supporting the GMP manufacturing of a blockbuster drug. So congratulations. That is very exciting news.
Thank you, Dennis.
Absolutely. So with that, I will transition to the topic of today's talk, which is not your grandparents' drugs, how drugs have evolved since the 1970s. I very much appreciated your partnership on this, Christian and Chris. Actually, not your grandparents' drugs came from, I think it was Christian's idea for the title, which I loved. So today we'll talk about one key property, which is lipophilicity. Are drugs getting more lipophilic over time? And this was a more interesting topic than I expected when we first started research on this. We'll go into that in a little bit. We'll take a visual look at how drugs have evolved since the 70s. There's some really profound things there, I think, and conclude with, what can we do about these drugs that are changing, and some solid form solutions that help make these drugs bioavailable. Okay, so first, just to start with a really basic concept, what is log P and why does it matter? Log P is one of these things that I think everybody joining the industry doesn't appreciate as much as we should when we first start. Certainly I didn't. I thought it was just one of these parameters that people measure just to measure. Didn't really appreciate the fundamental importance of it. So log P or log D is a measure of the partition coefficient of a molecule between water and a non-aqueous phase like octanol. This is helpful to get the sense of the lipophilicity or fat-loving nature of a molecule, or the intrinsic preference of a molecule to prefer a hydrophobic phase versus an aqueous phase. And why is this so important? Well, obviously in biology, you have this sort of phase separation all the time between cell membranes and water, between buffer solution in your blood and cellular compartments, but also within proteins themselves. So if you think about a molecule leaving water, a molecule that is more lipophilic is going to inherently want to bind to hydrophobic pockets in proteins more strongly. Now, because this is a logarithmic scale, something that has a log p that is 2 units higher than another is going to want to escape water, so to speak, 100 times more strongly than a molecule with lower log p. Now, where this is important in medicinal chemistry, drug discovery, and formulation is this is a universal, it's a physical property, right? So a molecule that wants to escape water and bind to a protein 100 times more strongly is going to want to do that for all lipophilic protein pockets as long as it's sterically tolerated. And so, where this comes into play is imagine you increase a molecule's potency by 10x. Well, if you're also increasing the lipophilicity of the molecule by 10x, you're not necessarily increasing a specific interaction. You could just be increasing the affinity of this molecule to all lipophilic pockets by 10x. And so effectively, you're not necessarily making a molecule that's more selective. You're not making a molecule that has a greater free drug exposure relative to serum protein. And essentially, it's a way that you can deceive yourself into making more potent compounds that aren't necessarily better drugs. So log P is essential for a lot of reasons. Everybody knows it's fundamental because you need to balance it for absorption as well. You need some passive permeability to get across cell membranes to be able to be orally absorbed. There have been a lot of studies looking at the log P of drugs over time, and it's sort of ingrained in medicinal chemists that the ideal log P of a drug is between 2 or 3. And there's this really famous paper from AstraZeneca by Paul Leeson and Andrew Davis in 2004, where they looked at drugs from before 1983, going all the way back to the 30s, and compared that to the cohort of drugs between 1983 and 2002, and saw that the mean value of lipophilicity, or log P, basically didn't change between these two sets of compounds and didn't really change over the decades. Now, they took this as evidence to suggest that, there's sort of a fundamental amount of lipophilicity, so to speak, that a drug should have, and that is generally between 2 and 3. So if you look at this bar chart here, you can see that the median change of C log P, between drugs from 1930 to 1983 versus 1983 to 2002 was almost 0. So when your team at Nanoform came to us and said, hey, we've been doing a lot of drug formulation. We've been noticing that there are a lot of challenges with modern drugs, getting them to be formulated properly. And you came to us with data showing that median log P values had been increasing over time. My first reaction personally was severe skepticism, because what you were showing was that the median log P of compounds in the last 20 years has increased by a log unit since, from drugs of the past 100 years. So are drugs 10 times more lipophilic on average today than they used to be? That would be a very dramatic claim. But we thought this was interesting, and so we decided to work with you to dig through the data a little bit deeper and see, well, okay, can we explain this? Because if we can explain this, then maybe it is real. So our first assumption actually was, maybe it's that we have more compounds that are greasier. So really, really lipophilic compounds with log Ps over 5 or 6, like everolimus, as you mentioned. That's plausible since molecules are getting bigger. We're seeing more arrow rings, you're seeing more, cross-coupling methods being used. Maybe we're just seeing a lot more lipophilic compounds. And actually, when we look at this data set, we saw that surprisingly, the number of compounds with a C log P over 5 has actually decreased in this time period. So the shift in median log P can't be explained by just an increased number of bad actors. Also, if you look at this middle section, this purple group, if you look within that, the average compounds there aren't actually getting much more lipophilic. That has stayed relatively constant over the years as well. Surprisingly, what has changed is that the group of compounds with log P below 0 has actually decreased significantly in the last couple of years. So you can see from 1990 to 1994, about a quarter of drugs had log P below 0. But in the last couple of years, that's actually reduced to anywhere from 8 to 13%, so about half as many drugs. And so if you look at the median log P of approved drugs overall between these time periods, or the mean, it's increased, by almost a log unit from 2.3 in the 90s up to 2.9 to over 3 in the last 2 decades. So this was a really surprising, unexpected finding. So it does look like the median drug is getting more lipophilic, but not necessarily because the drugs themselves are getting more lipophilic, but because overall the populations are changing. So this led us to ask, okay, so what happened to all of the polar molecules? And we dug into the structures to see if we could find some trends. And as a synthetic chemist, it just sort of hit us as, oh, duh, guess what has C log Ps below 0? All these natural products that have gone away. So as many folks in the audience, I'm sure you have the same reaction, where we all know that in the last 2 decades, pharma and biotechs have shifted away from natural products as inspiration for new drugs. There have been several recent reviews talking about this. There's a great 2009 Science paper actually highlighting this phenomenon. And it's sort of general knowledge that natural products over the last 15 years have essentially disappeared from the pipeline. Whereas from 1981 to 2010, about 50% of drugs actually had natural products as starting points. So that was kind of profound. And it was really nice to see, after we had published these articles, team at Merck, led by Ingo Hartung, had explored this as well, and they found also that drug lipophilicity has been increasing significantly, both in the oral set and overall. So this was really nice validation of this work. So from there, it was really cool to go back and look then at only the synthetic drugs from the 1970s and see how synthetic drugs have evolved over time. When you mash all these things together and you include natural products in the analysis, ring size, ring count, hydrogen bond donors, acceptors, right? It's really hard to tease anything out from the noise. But if you just look at the molecules and only the molecules that came from chemical synthesis, you start to see that, yeah, the drugs from 1970 don't hold a candle to the drugs of today. Even by 1990, the typical drug maybe had 3 or 4 rings at most. These are probably the most synthetically complex molecules that were fully synthetic, only 30 years ago. But if you look at the kinds of drugs that are being approved today, just a few decades later, I mean, we've got molecules that are present as single atrope isomers like sotorasib. You've got molecules with 6, 7, more rings. Atogepant is an interesting oral drug for migraine that's got 6 or 7 6 rings there. It's got a spirocycle with stereochemistry. It's got 3 stereocenters on that pipridone ring. Lenacapavir has 10 fluorine atoms. And we've even had a deuterated drug be approved recently with deucravacitinib. So just on visual inspection, it sort of now is much more strikingly obvious that the drugs of today are very different from those of the past. And I think with the advances in synthetic chemistry and drug discovery technology overall, and the economics of how much we're able to outsource and create molecules at a faster pace than we ever have been before, I think this trend is only accelerating. I mean, as we look at molecules that we've highlighted in molecules of the month or molecules of the year, there are some truly mind-blowing structures. This compound on the left is a GLP agonist molecular glue from Eli Lilly that we highlighted, I think in 2022. This molecule has, this crazy cyclopropane on the left. It's got, 9 or 10 rings. It's orally bioavailable, wild structure that you wouldn't have seen 20 years ago, or even maybe 10 years ago. We have obviously molecular glues and protacs and degraders, like the molecule on the right from GSK. Now we're starting to see oral bifunctional molecules and even brain penetrant bifunctional molecules, which is really incredible. We covered 2 recent conferences, ACS at Indianapolis and AACR in April. And it was really astounding, again, to see how much chemistry has transformed what we're able to do with small molecule drugs. This compound on the left is the first oral PCSK9 inhibitor to be successful in the clinic. It actually has an oral bioavailability of less than 5%, and formulation in that story was really critical to getting the overall drug properties and absorption needed. So I thought that was a really interesting example. That compound is over 1,500 molecular weight and is really a tremendous accomplishment by Merck. These 2 molecules on the right are, this one in the middle is a degrader from C4 Therapeutics. Again, this thing has 9 rings, but it is orally bioavailable. I think its dose is around 30 mg once daily or something like that. And it has the potential to revolutionize the treatment of resistant BRAF-driven cancers. This compound on the right, again, is a natural product, remotely natural product inspired glue-like molecule that actually recruits cyclophilin A to KRAS while covalently binding KRAS. It's a really interesting new modality for drugging a target that has been considered undruggable for many, many years. So I think it's really cool to just look at these molecules and recognize, how much fundamental chemistry has, again, opened up so many doors for us to create new products beneficial for society. So with that, with us moving into this uncharted territory of, over 1,000 molecular weight drugs, compounds with dozens of hydrogen bond donors and even acceptors, increasingly solid form solutions and other non-traditional solutions are becoming important to getting these drugs into the clinic. As I mentioned, the MK0616 molecule has a oral bioavailability of less than 5%. And the right formulation was essential for getting that drug to its 30 mg once daily dose. So with this, I'll hand this over to Chris Worrall. Maybe you can talk us through what are the solid form options for making small molecule drugs have more ideal properties, and tell us a little bit about your technology.
Yeah, sure. Thanks, Dennis. Yeah, I mean, obviously, as you said, we can go through now and create amazing molecules that can do fantastic things, but at some point you have to be able to get them into the body, and obviously into the areas where they're actually going to have the desired effect. And so there's a variety of different things that we can potentially look at. If I start maybe with what's been historically the most common in terms of salt formation, right? I mean, maybe as a med chemist, we go through and make a molecule, and it doesn't quite have the solubility that we need. It has a basic group, so okay, let's make the HCl salt and go forward. And so, that's perhaps been the most common and maybe the most widely understood of the opportunities to improve on solubility and on dissolution rate. But obviously, there's a few drawbacks from that. One, obviously, being the fact that you need an ionizable center in order to do that. And obviously, as molecules become more complex, the possibilities of having numerous ionizable centers with similar pKa's obviously increases and potentially makes that more challenging. And then there's also the fact that when you form a salt, you are likely to vary the hygroscopicity. And certainly, again, when one thinks about sodium and HCl as the most common salts, they often have issues with not only hygroscopicity, but also with hydrate formation. And so again, being able to control the crystalline form and then the hydration states is obviously going to be critical from that perspective. And of course, we're adding molecular weight. Now, that may not necessarily be as much of an issue for something that has 1,500 molecular weight, but might be when something is only a few hundred. We also have particle size reduction approaches, right? And so there, if you can increase the surface area of the particles, you can get better wettability.
therefore improve the dissolution profile and potentially improve bioavailability. We can maybe get faster onset. Of course, some of the issues with particle size reduction are that by definition, you're breaking particles. And so you're potentially introducing defects within the crystal lattice. Of course, as the particles knock into themselves, into the balls that are in nanomilling, for example, into the surfaces of the vessel for jet milling, then potentially you're changing the surface properties. And so we might get small domains of amorphous that can change things such as the cohesive forces and can potentially lead to agglomeration over time after having gone through those particle size reduction approaches. We can also make use of amorphous, but obviously the issue with the amorphous is that it's the highest energy level state, and so it has a tendency to want to crystallize over time. There's also the potential that actually it's less chemically stable as well than crystalline material, and so there one might look to do a dispersion, obviously, where you can keep the molecules apart so they don't crystallize, and it can also help excuse me, to reduce the potential for chemical degradation. But of course, one of the key issues with that is we're adding 50% or more of a polymer to our API, which will reduce the overall drug load that we can get within our drug product. And so that can lead to maybe multiple tablets or larger tablets, which may or may not be amenable to patients. Certainly those undergoing chemotherapy, for example, issues with dysphagia, struggle to swallow, may not be able to take large or multiple tablets. One of the other potential issues with spray drying in particular is the vast amounts of organic solvents that are required. And of course, we're often talking about less than ideal organic solvents that are used in those processes. And then we have other things, like such as co-crystal formation. Right? And so there, one of the potential advantages over salt formation is that you don't need an ionizable center. But again, you're potentially adding significant molecular weight to the API. And of course, you actually have to go through and do quite a lot of screening in order to identify potential co-formers that will form the co-crystal. And there's actually not that many reliable ways to predict the properties of the co-crystals that you're going to get. And so you can go through and make a whole range of co-crystals that actually may not necessarily offer any advantage over the parent form. So I think those have kind of been historically a lot of the methodologies that people have used. And then, we can come on to CES, which is Nanoform's technology. So that's controlled expansion of supercritical solutions. And so we use carbon dioxide as the key solvent for our process. And we essentially do a nano crystallization process. So we can generate crystalline nanoparticles of the API directly from solution. So we can have dissolution advantages, obviously over the parent form. Potentially, we can also see dissolution advantages over salts and co-crystals, and I can come on to that in a second. But we also have advantages over particle size reduction because it's a direct crystallization process. So we're not changing the particle properties when we reduce the particle size. We're not changing the surface properties, and so potentially we can see less issues with agglomeration over time because it's a direct crystallization process. We can also potentially tune the particle size distribution that we get as well. And so some people may want the smallest possible particles they can get in order to improve oral bioavailability. But if one thinks about pulmonary delivery, for example, you may want crystalline particles in the 1 to 5 micron range. And actually, if we can go through and directly crystallize from our process like that, again, we can potentially have much better properties when we go into a dry powder formulation than you would have from a particle size reduction approach. Great, thanks, Dennis. And so, yeah, I mentioned before in terms of the improvement of dissolution. And so this is some work that we did actually in collaboration with the Pharmorphics group, where we compared nanoparticles generated by our CES technology versus a whole range of other particle engineering approaches that one might look to take. And the example here is for piroxicam. And so what we saw is that we saw a faster dissolution rate and a higher apparent solubility out to 30 minutes than all of the other approaches. So you can see here we have a salt, we have a co-crystal, we have milled material, micronized material, and then also a dispersion by spray drying and by hot melt extrusion. And so, we certainly, for us, it was very interesting to see that actually we could potentially outperform dispersions without the requirement for significant volumes of polymer in order to be added to that. And we can see that we can also outperform salt and co-crystal formation just by the reduction of the particle size. And in this case, as I mentioned before, this is nanocrystalline material. So it's a direct nanocrystallization process of peroxicam. And this was actually also borne out in clinical data. So the first clinical data that we can share with the phase 1 peroxicam. And so what we saw was a 33% increase in the AUC in the first hour compared to micronized material. And so clearly that's, we're thinking about patient centricity and faster onset, potentially for pain, migraines. If we can get a faster onset, then obviously that's going to be beneficial for patients. And we saw a reduction in the Tmax for our product compared to both micronized material, but also compared to beta-dicyclodextrin coupled material. And so both of those were marketed products. The micronized is Felden, which is a product from Pfizer, and the beta-cyclodextrin coupled formulation is Brexidol, which, as I mentioned, uses beta-cyclodextrin to improve that. And so we actually saw a reduction in the Tmax compared to both of those marketed products by the generation of nanoparticles using that process.
All right. Thank you, Chris. This was something I didn't really appreciate before thinking about this. One, it's, we appreciate the explanation, like why nanocrystalline material would be better than amorphous, because you kind of have all the benefits of, the amorphous faster release with having actual stable crystalline material, which is really great. But I think for medicinal chemists, one thing that's really valuable here is the fact that you can modulate the product properties like the Tmax, right? I mean, that's tremendous. For example, if you're trying to differentiate in migraine and you want a faster acting drug, or in insomnia, for example, where 30 minutes or something like that of pain would really differentiate your product, being able to tune that with your solid form could be very powerful. But it's also powerful if you have a drug that can be nanoformed, but somebody else's can't. That could be, really a differentiating factor. And I imagine that you must be talking to people in like the CGRP space, for example, on how to do that.
Yeah, and as you said, if we can go through and change the properties of the API first, if you can start from a higher level, when you go through and do your formulation, potentially you can see even greater improvements. And certainly one of the things that we have seen is that it isn't just the generation of the particles, it's what you do with them as well. Mm-hmm. And so making sure that you have the right formulation, you can really get the best out of the particles that we produce, and really see those significant changes. And yeah, as you said, anything, I mean, that's just one example, right, where faster onset could be significant improvements for patient benefits. But actually, one of the interesting things that certainly we found has been that the utility of nanoparticles isn't just for oral. I know that's obviously the specific what we're talking about here, but we can potentially use them to enable a whole range of different drug delivery approaches. And so, for example, one can think about long-acting injectables or polymer implants. And actually, one of the interesting things that we've seen is that when you reduce the particle size with the same mass of material, you can actually see a longer therapeutic window. Mm-hmm. Now, that seems a little bit counterintuitive, but if the rate of the release is not dependent on the biodegradation of the polymer, mm-hmm. Then it's the diffusion of the particles within that polymer matrix is going to be the key factor. And so if you think about, distance of travel, for example, and the size of tunnels within those polymer matrices, larger particles have relatively less distance to travel, and the tunnels within that polymer matrix are also slightly larger. And so potentially you can see that diffusion is slightly faster. And so hence why we actually see a longer therapeutic window for nanoparticles than we do for slightly larger particles. And so again, you think about patient centricity. If you can elongate the therapeutic window such that you only need to be dosed every year, as opposed to every 6 to 8 months, then obviously that's going to be beneficial.
Yeah, this is very interesting because as you think about the drug life cycle, I'm sure everybody's wheels are sort of turning internally now. How can you continue to add value with with a drug that can have all these enhanced properties? This really becomes very powerful clinically, commercially, and IP wise. I imagine these nanoparticulate formulations provide another IP angle as well, don't they?
Yes, indeed they do. Yeah, and as you said, we can use it either at an early stage to enable more molecules to make it through, as Christian mentioned before, potentially refocus the lens and give us a wider selection of compounds that we can actually look at and look to take forward. But then, yeah, as you said, life cycle extension as well. And so, yeah, we can certainly add additional intellectual property surrounding the molecule, and of course surrounding its dosage form as well.
So yeah, for medicinal chemists, I think this becomes very interesting, where now you've got, let's say, 2 clinical candidates. One has the potential to be nanoformulated, one doesn't. I mean, it sounds like it's a no-brainer then to go with the one that has the properties that are potentially enable these different solid form technologies. So you mentioned before you have a tool, StarMap, that helps predict this. Can you tell us a little bit more about that?
Yeah, sure. So we do indeed. So it's a sparse data AI platform, which will take information about the molecule. So at a bare minimum, just the SMILE string is the minimum that we need. But if there's information on the melting point, the enthalpy of fusion, obviously log P, we've discussed a lot. All of that will just help to improve the accuracy of the prediction. And essentially, we predict 2 key parameters for our process. The first being solubility in supercritical CO2. The second being the propensity of the molecule to crystallize under those conditions. And so what we can then do is we can plot those on basically on a matrix system. So we have solubility on the x-axis and propensity to crystallize on the y, and we can then rank either a whole range of compounds to see which looks to be most amenable, or if there's only 1 or 2, we can see not only which looks to be most amenable, but that can also help with our initial process development work as well. And so that can actually help to drive the process development work and really focus in on what experiments we need to conduct in order to get the fastest process development work. So it works in 2 ways, basically. And as Christian mentioned, we actually have an online portal which is available to our clients, that they can go in and submit molecules themselves and get the StarMap ranking for those in an automated fashion.
Well, thank you, Chris, for sharing. Christian, before we turn this over to the Q&A session, would you like to add anything?
I think it's been a really interesting discussion, and clearly drug molecule complexity is only going in one direction. We've seen that over from our grandparents' drugs, and I'm sure our grandchildren's drugs may look even more complex as we go forward. But I hope that this session has provided hope for those that have tuned in to realize that there are technologies available to help address and to make these molecules more patient centric and available to patients first and foremost. And I guess just one parting thought: small is an ingredient in formulation, but it's not just about particle size. Making particles is one thing, but we have to get the value out of those particles, and coupling it with the right formulation strategy is really important. So Nanoform, we've invested a lot of time and effort into understanding the physical properties of the particles, to characterize them, to really understand what we're making, because as we're pushing the boundaries of science into making these incredibly small particles, down as small as 10 nanometers, we need to be able to also push the boundaries of analytical science to fully characterize them and understand what we're making. But we also are challenging ourselves to make sure that the formulations can deliver the value that these particles can potentially offer for patients. So it's really a combination of understanding the challenges around processing and making a particle, understanding how to characterize it, and understanding how to formulate it to get the right result in vivo. and that's something that we're working diligently on with our partners. And of course, we'd be delighted to work with new partners and obviously increase our existing relationships.
Thank you. Well, thank you both for sharing. I think as a former medicinal chemist, it's really fascinating to hear this, new dimension really to solid form that hadn't been considered before. I hadn't even thought about the fact that you can now combine existing formulation strategies, with this particle size control to now, get a lot of new profiles, tunable profiles as well.
I mean, Dennis, if I may just mention, we had a huge success last year with a company based in Portugal called Targtex, where they're looking at glioblastoma treatment. And they had a hydrogel based approach for patients that come with glioblastoma, and effectively they couldn't get enough drug load into the hydrogel. So this wasn't around necessarily bioavailability, but it was a formulation challenge, and they realized that particle size was key, and they tried nanomilling, and it increased the drug load by 40-fold. And so that was good. The rats, the cancer, basically the cancerous cells disappeared, but then they all came back again, and the rats eventually died. They came to Nanoform, and we increased the drug load by 200-fold. 40% of the rats just were kept alive till the end of the study, and we went up to 160 days. And we had a subtherapeutic dose on this. So it's not just about patient centricity, making a slightly improved formulation. It can be life-changing. It can provide hope for patients with these terrible diseases. So that is a great example of where a smart formulation coupled with nanoparticles can have a tremendous impact for patients. And we're really excited. And that's one of our GMP partners, one of the drugs we're also working on.
Interesting. Well, it's absolutely fascinating. I think the longer we spend on this, the more applications you start to think of. I mean, you mentioned, I hadn't even considered pulmonary applications, right? If you have an inhaled drug, how do you maximize, I think that's a big problem in IPF, for example, just getting that exposure with the scarred tissue is very challenging. Actually, you mentioned this rodent study, actually just getting proof of concept done with a small molecule, like let's say you can't get the exposure that you need just to see if you're having the effect. Why not try a nanoparticle formulation of your early compound to make sure that you can do all the studies that you need without making new compounds? So I love it. So really appreciate this this very inspiring sharing of this new technology. Thank you.
Thank you, Dennis.
Absolutely. Well, with that, I guess we can turn it over in the last 10 minutes to questions from the audience. The first question, actually, these are for you. I'll start with the questions for you. One is from Sean Walker in the US. For the CESS technology to generate nanocrystals, do you quantify the percent crystalline versus amorphous content in the nanoparticles contained? And what are you typically seeing?
Yeah, that's a good question. Obviously, that's obviously going to be one of the key things. And yeah, I mean, the short answer is yes. We have done some of that work to go through and quantify, and we are seeing that we are generating highly crystalline material. I don't know that I have any specific examples that I can share in terms of the percentage from any of the work that we've done, but we have looked at that in a variety of different ways. So, I mean, obviously, XRPD is one. DSC being another. We've also done some work looking at IOEM as well to go through and look at that, and that's not only looking at the bulk, but we've also gone through and looked at individual particles as well to say, okay, how do we know that each individual particle is crystalline? And so we've done some of that work as well. So looking at both the bulk and individual particles to confirm that we're getting consistent crystallinity within the material.
Thank you.
Yeah, I guess just to add to that, sometimes we can make crystalline nanoparticles that are sort of down to maybe 100, 200 nanometers, but we can't go smaller than that. It depends very much. Crystallization is very much driven by the molecule, but we can make amorphous particles that are smaller than that. So we've actually developed a new formulation platform that we're not going to go into detail on today, where we can, when we've patented this, where we can actually convert the amorphous nanoparticles without changing their size to crystalline nanoparticles, and that's hugely exciting. We'll have maybe an article on that sometime soon with you, Dennis.
Yeah, absolutely. So coming back to our earlier questions from the talk, we have from Thomas Babinski, lipophilicity is one thing that's well addressed in formulation. What about insolubility due to pi stacking disruption? I'm going to just quickly, I guess, comment on our data. We did look at the fraction sp3, for example, of the molecules over time. I think just visually looking at the molecules, obviously, they're getting bigger, more three-dimensional, more complex. So I think that may be counterbalancing, some of the issues with these molecules getting bigger. But I think, that pi stacking disruption and other ways to increase solubility are certainly things to consider. I don't know, Chris or Christian, if you have any thoughts on that before we move on.
Yeah, I mean, I'm not specifically on the pi stacking as such, but obviously in terms of crystallization, one of the things that actually we've shown on a couple of occasions, and atovaquone is one of those examples where we've actually been able to control the morphology of the material. And so the thermodynamically stable form of atovaquone actually prefers to form rods. And so, whatever the stacking is, if you imagine pi stacking, you're probably going to get that in one direction. And so you're likely to lead to particles with a larger aspect ratio. And actually, we were able to show that when we generated particles in the 300 nanometer range, we were able to change the morphology. And so to move them more towards a sort of spherical shape. And so to significantly reduce the aspect ratio of those particles. And so again, being able to potentially do that could be very beneficial, not only from the bioavailability perspective, but also from a formulatability perspective as well.
Gotcha. All right. Next question is from Dilani Fernando. She asks, has anybody done the same study for all molecules taken to the clinic and its survival to market? Off the top of my head, I'm not sure. I'm sure somebody probably has attempted to do a subset of this or some analysis. I do think it would be fascinating, and maybe Christian and Chris could be part 2 of this series.
I think the challenge there is what's disclosed in an early stage. From a chemical structure perspective is one aspect. So to really understand which molecules failed, but the biggest challenge is to understand why they failed. There's very little reasons given for that. And I think scientifically it's great to report the good results, but it's perhaps just as important to report the bad ones so that we can all learn from a scientific perspective how to develop better science and ultimately better medicines. Yeah, that would be fascinating to really understand all those molecules that failed in phase 1 or in phase 2 due to bioavailability issues, what the lipophilicity was like for those compounds.
That's a really good point. Yeah, there is some survivorship bias in this analysis for sure. So Fernanda Cerquero asks, if natural products were revisited using the new nanoparticle formulation techniques, I guess this kind of gets to your point, could we find drugs that had been neglected before? And I'm sure you have already looked into this, but.
Absolutely. I think there's a treasure trove of molecules that could be taken forward. A lot of companies have these war chests of molecules. The challenge is, from our perspective, is trying to get partners to reevaluate molecules that have failed, right? Because nobody wants to bring back something that they had to make lots of decisions within the company to kill, to bring it back to the table and to try and give it another chance. There is definitely a reluctance to do that, but we would be absolutely delighted to explore molecules that could be given a second chance to see whether we could make a success of them.
Sounds like there's an opportunity for an entire biotech to start in this area. Maybe something like Roivant, Nanovant, maybe for nanoparticle formulated drugs. Could work.
You heard it here first.
That's right. Should patent the idea. All right, so Paul asks, an aside curiosity historical question for Christian. Sonochemistry continues to be popular and useful, but promising and reliable sonocrystallization technology usage appears to have faded. What is behind this trend, and is it an IP or patent issue, expense, scalability, or some other reason?
That's a great question, and one obviously that is close to my heart. And I think was very passionate, and still am very passionate, about the use of ultrasound for crystallization. I saw the advantages that it offered for many partners that Prosonics worked with many years ago. Unfortunately, a subject of, I guess, the way that company then progressed was they were acquired by Circassia, and Circassia had a phase 3 pollen or allergy product that failed in clinic, and that company then unfortunately sort of didn't succeed and didn't go forward. So all that IP that resided then in the parent company Circassia then sort of went away, wasn't protected further. But I think, you know, the technology works. It works very, very well for seeding, nucleation, control of crystal growth of particles, for making different types of particles. And, you know, I would actively encourage people to use it. I don't believe there's much IP protection anymore around the technology, but it's a very, very useful tool. And I think it has great merit still.
Cool. All right, well, we'll conclude with our last question from an anonymous attendee. What's the possible scale of applying this nanoformulation?
Limitless. So it's a great question. And as I started off at the beginning of my talk explaining my background, and when I came into Nanoform, being perhaps slightly skeptical around scalability, reproducibility, I was delighted that Nanoform convinced me otherwise. But the true throughput of the technology, and we define scale by throughput, is defined by the molecule itself, and that's why we measure its solubility into carbon dioxide. So something that has a very, very limited solubility in carbon dioxide will have a much lower throughput, and something that has a higher, obviously, will have a higher throughput. So that's why we use StarMap to assess the technical fit of the molecule with our technology, to take it forward. And if it's, you know, a large volume product that's, you know, hundreds of tons, but the solubility is very limited in carbon dioxide, it's probably not a very good fit. But if it's a product, you know, that's sort of in the tons range, but it has a higher solubility in CO2, it could well be a great fit. You know, and I've seen the technology progress in the last 5 years since I've been in the business, 4.5, nearly 5, from looking at, you know, talking about milligrams an hour through to hundreds of grams an hour, and even potentially kilograms an hour for certain molecules. So I've been very, very encouraged by the scalability that we've shown, and the expansion of our GMP capability. So I guess one thing we didn't touch on, but we have this GMP expansion that we've done in Helsinki. We've now got high potent capability to handle oncology molecules in the sort of 30 nanograms per meter cubed OEL, from a safe handling perspective. And we're looking to expand into the United States, to have a GMP manufacturing facility there too. So, you know, as we do all of this, the technology is constantly evolving. And as you quite rightly pointed out, Dennis, yesterday we had a press release around renewed license from our Finnish Medicines Agency and commencement of a blockbuster marketed drug.
All right, thank you, Christian. All right, well, I guess we have one last late-breaking question, maybe give you 20 seconds to answer this. Do you have any thoughts about adapting your technology in the use of oral peptide formulations? I'm curious about this as well.
Great question, and thank you for that. So in actual fact, we have 2 technologies. So what we've spoken about today is our CESS technology. It's founded on supercritical CO2, as Chris pointed out. But in the last few years, we've also developed a new technology for biologics. And those CESS typically goes up to around about 1,000 molecular weight, maybe 1,200, 1,300, maybe into that small sort of peptide range. Our bio platform, which is also solution to particle and doesn't necessarily use supercritical CO2, so aqueous based, this technology can go from 1 kilodalton up to 150 kilodaltons. So we started to look at monoclonal antibodies. We've looked at molecules like insulin, making nanoparticles for drug delivery. There it's often perhaps less around bioavailability, but more around, increased drug load per unit volume, tailored release profiles, making dry nanoparticles of biologics, which is typically an IV route of administration. Thinking, what does that open up? Can we do nasal delivery? Can we do inhaled? Can we do, dermal applications, ophthalmics, etc.? So yes, absolutely. We are very much in the peptide space, and we'd be delighted to have a conversation about that.
Awesome.
Thank you.
Fantastic. Well, thank you, Christian. Thank you, Chris. Thank you, everybody, for attending. And with that, we will wrap this session. Thank you very much for a very fascinating talk.
Thank you.
Thanks, Dennis. Thank you, everyone.
Absolutely.